It says I've got a good connection, so.
Okay. Well, I'll just go on here.
Yeah. Okay. Can you see?
Okay.
Can you see us here?
Hey, folks.
All right. There we are. Hold on,
You guys
Steve.
looking a bit pixelated today.
Okay, so we're recording as well. Take it away.
Okay. We're going to talk about, we're not going to talk about the case on paper today, that'll be Thursday. Today we're going to talk about some of the preliminary stuff, life and ecosystems. And what I'm going to try to present to you is sort of the theoretical way we think of both life itself in terms of cellular life and ecosystems, which are the interacting system of living beings and organisms of various kinds. We're going to start with a little chemistry though, because if you're going to try to understand life itself, in other words, how a cell works, and typically when people talk about life, they're referring to a cell. Got to do some chemistry, because basically that's what life is. It's a collection of chemical reactions. So, here's some notation. You may have seen once upon a time chemical reaction written like this, where you have A plus B goes to C, where A and B are called the reactants, they're the things you start with. C is called the product, the thing you end up with, and this arrow is called the yield sign, and it's often pronounced simply goes to. So this is like I mix A with B and I end up with C. Now, another thing that happens, and another kind of chemical boogeyman that we have floating around here is, and we write it like this, we put a little letter over the arrow. D here is what we call a catalyst. A catalyst makes a reaction run faster, or more to the point, okay, a catalyst controls the rate at which a reaction goes. So think about it this way. For if you had just a solution with A and B in it, A and B would have to float around that solution and collide at random, okay? Not only would they have to collide at random, they would have to collide with enough energy, and they would have to collide in the correct geometry, the correct orientation. Now, the reason chemical reactions happen is because we have billions of collisions per second going on in the beaker. However, if you do this with a catalyst, okay, what it is think of it this way. If you're trying to meet somebody in a crowd, it's hard, okay? But if you know your friend likes a certain coffee shop, you go wait at that coffee shop and your friend might show up. That's exactly how a catalyst works. It's a place where A and B can meet, okay? Where they can meet, and given the way they fit into the catalyst molecule, they're held in the correct orientation. So they can make a reaction go a lot faster. Now, if suppose we had a whole series of chemical reactions, so A plus B go to something else, goes to something else, goes to something else, goes to something else, so on and so forth. But at some point, the critical thing, you make D. D is one of the products. D is also the catalyst for this first step. So we call this first reaction autocatalytic. In other words, at some point, at some future point down the line, this series of reactions produces a catalyst for at least one of its steps. Okay. Now, autocatalytic reactions are critical to living systems. The reason is you can control this reaction very carefully using feedback. In other words, if D speeds up the rate of this reaction, okay, you can do something down here to inhibit the rate of production of D, which slows this reaction down, so on and so forth. So in other words, it gives you a way to reach in and control this reaction. Now, biochemical catalysts are called enzymes. And what you have to realize here is that if A and B are regular kind of chemistry molecules, they have anywhere from, let's say, 3 to 50 atoms. Okay, even in organic chemistry, the molecules get big, we're talking 50 atom limits. Biomolecules, we're talking thousands, maybe even tens of thousands of atoms. We're talking about things that are much, much larger. And they're still technically molecules because they're molecular level objects. But the trick with a catalyst is that it's best almost thought of as a nanomachine. I always laugh when I hear people talk about, will nanotechnology be possible? Of course it's possible. It's called biochemistry. You know, what you want to say is, will we ever invent our own kind? God already got his out the door. Okay? And catalysts are huge molecules that slowly over time have evolved to react in certain ways. They change shape, they hold on to things, but they can also let things go. So biochemical molecules, especially enzymes, can be thought of as little engines, okay? They can literally take the random motion that's going on at the molecular level and convert it to work, which is what an engine does. Now, remember something really interesting about life at the molecular level. These things are always moving. There's a ton of energy down there. You don't have to make them move. What you have to do is make them move the way you want them to. So we're not looking at this heat reservoir or cold reservoir. What we're looking at here is little molecules that act as engines, okay, that literally take the random motions they naturally experience and organize them to some extent to be able to do work. Now, there we go. This is kind of an alternate notation that's used in the in certain literature, and is using the one I want to know. So notice I typically write a chemical reaction like this, and you've probably all seen this in high school, maybe even a college course. Okay, here is a different way to write that same reaction. Here, the reaction itself is called reaction number 1, and it's represented by a little circle. A and B go in, they have solid lines, and C comes out. So it's very similar to this, but we just actually, rather than use the arrow, we just use a little dot.
Yep, at least I do.
That's just a little bit. Okay, now, a wordy slide, always dangerous. Okay, what is the most general definition of life we currently have? Well, this is simple. If you have an autocatalytic set, okay, what is that? An autocatalytic set is a collection of entities, each of which can be created catalytically by other entities within the set. And by entities here, I mean some sort of molecule, such that as a whole, the set is able to catalyze its own production. So in other words, you've got a collection of chemical reactions which not only create its own catalysts, but eventually can create every molecule that composes that set. Okay? So in other words, if you start off with a little bit of this stuff and you just let it go, eventually you'll end up with a lot of that stuff. Okay? Everybody see what I'm getting at there?
So it's sort of like self-organizing, developing?
It's not so much, it is a self-organized thing, but mostly if we're talking about once it's there, what you have is a set of chemical reactions, and the only critical thing here is that within that set, they produce a catalyst for every reaction in that set. Okay, so in other words, they produce their own catalyst, and they produce everything else in that collection of molecules, so that this thing can basically reproduce itself. Okay, and that's what we call an autocatalytic set. We actually have programs that can mimic this, autocatalytic sets that replicate themselves. Okay, so for example, what you can have is, you take this autocatalytic set, let's suppose you have a solution that's running these reactions. You can pour half the solution into another beaker, and both those beakers will now be running that set of reactions. Okay, so as long as you don't reach in with this good tweeze and pull out specific molecules, simply dividing the solution in half gives you gives you exactly two things. It'll just keep growing on their own. Now, here's the definition. NASA defines life. Notice it's NASA. They need a very broad definition because they're looking for life on other planets. We can't guarantee that it's going to use nucleic acids or proteins or anything. So NASA broadly defines life as a self-sustaining chemical system capable of Darwinian evolution. Autocatalytic sets fit this definition. So it's a self-sustaining set of chemical reactions, like I described up here, capable of evolving. Okay, now, that's a key word. I've done a lot of chemistry up to now, but this clearly brought biology back to the center. So, here's a little biology. Okay, we're going to define what Darwinian evolution is. One thing you have to realize about life from a scientific perspective, something like a cell or a virus, it has no goal, it has no purpose. What it does is survives. Okay? If you survive, you're a successful life form. That's all it means. If you're not a successful life form, you die. Okay? Without any making cell. One of the best survival strategies that life on planet Earth has discovered is replication. Okay? If you make 8 copies of yourself, it's a lot harder for someone to come along and wipe you out. Just by accident. There's only one of you, you're doomed. Okay? It's the first problem you have, and there's no more of you. If there's 8 copies of you, it's a lot harder to get you to take you out. Now, this gets even better if you notice that a collection of copies of yourself is what we call a population. So it's a collection of people, a collection of frogs, a collection of cells. And this works better if these populations can evolve. And what does that mean? Well, here, it means within this population of self-replicating entities, okay, up here, you have to be able to have small modifications occur at random. So in other words, some cells do this, some cells do that, and it happens randomly. So you end up with, initially, the replication is perfect, or roughly perfect, or almost perfect. But there is a small variation that arises, so these are all slightly different things. Okay? So you need to, and this is a hard balance. You need to process it as a faithful enough replication process such that it doesn't produce many errors at all, but it does produce some in very small amounts. Then, the other feature of Darwinian evolution is selection. So suppose the environment changes in some fashion. Something has happened, a new predator is in, the weather has changed, the food source has gone away, something. Within your population, some of the entities, some of the little critters, are better suited for this new environment than others. Just randomly. In other words, if if the change favors objects that are a little bigger, then the slightly bigger ones will have an advantage over the smaller ones. And sometimes it works the other way. Okay? Because of that, the ones that have the better variations, that have the favorable variations, have more chance of replicating. And eventually, those entities will simply out-survive the others and dominate a modified population. So if you have a population where tall things are favored, then you'll end up with tall ones. A good example of this, not in terms of molecules, but in terms of critters, is swallows out in California. These little birds have taken to living in overpasses. Okay, so you're on the highway, okay, and you go in the overpasses all the time, the two highways cross each other. Well, birds love to live, these little birds love to live in the overpasses. They also feed on insects in flight. So in other words, they catch flying insects while they themselves are flying. Well, a strong natural selection occurs if you live in overpasses, and they're called speeding cars. Okay? These little things would be chasing a bug, and if they weren't careful, they get smacked by a car, and that would kill them. And what's interesting is biologists have been tracking these populations of swallows for 75, 80 years. And remarkably, they saw evolution. What happened is they evolved to have a population of shorter winged swallows. Because within any, one of the great variation points in the selection of birds is how big are the wings. Some birds have bigger wings than others, even within the same species. Well, it turns out birds with shorter wings were more maneuverable. And they were able to get out of the way of the cars faster. And since the cars came flying down that highway every day, this was a strong selective pressure. And they literally document this, a very rapid change in the structure of these little birds.
So I want to point out.
Questions about any?
Well, yeah, Steve, I just, not really a question on my part, I just wanted to point out for the class that this is not the first time that we've encountered sort of the importance of difference and novelty, and it won't be the last, for sort of pushing a system forward and allowing change to happen, new levels to emerge, new better efficiency in processing energy or information. So, any other questions? Okay. Yep.
Okay, this is awful, but it's actually not that complicated. I'll explain everything. This is a model of what what they call a catalytic reaction system would look like, okay? And the letters A, B, C, D, G, E, F, so on and so forth, all represent molecular species. The R's represent specific chemical reactions that run. Okay, and there's 4 of them, 3 of which are catalyzed, 1 isn't. And then there's a piece of this whole thing that is autocatalytic. Reactions 1 and 2, including molecules A, B, C, and D. That is an autocatalytic set. Now, I realize I'm just giving letters here instead of real molecules. But let's be honest about things. If I started calling these, I've just used the chemical names, it wouldn't make any more sense. Okay, we got to see this. So this is an example of a very simple system. You can do this. So let's actually peek at it. A and B are food. They come in from the outside. All biological systems need food. G goes out. It's waste. Any kind of waste you want. It could be liquid waste, solid waste, gaseous waste. A, D, and F also can act as enzymes. Okay, so there's D, there's F, these can all act as, and A. It's not at all unusual that a lot of the food you eat can act as an enzyme. And finally, E, right here, is a plain old, don't mess with me, intermediate. It just means from going between reaction 4 to reaction 3, C and D produce E, and that just changes into G, ultimately. That reaction, R3, is uncatalyzed. What that typically means is this reaction proceeds pretty fast on its own. And it doesn't need any regulation, so it always happens quickly. If you want an example of an unregulated reaction that happens quickly, light a firecracker. Okay? You don't have to encourage a firecracker. Once you've lit it, you want to get rid of it quickly because you know it's going to react without much problem. Now, let's look at how this set works. A and B come in. So, they come in the presumed mouth of this species, okay? And they undergo reaction 1 to produce C. Okay? C is just some other molecule in the system. D is the catalyst, so this reaction is catalyzed. C then can proceed by reaction 2 to produce D. That is catalyzed by A. So this piece up here is an autocatalytic set of reactions. You keep feeding those things in and we'll run the circle. Now C and D can go through an uncatalyzed reaction, R3, because there's no need to catalyze it. It produces F and E. So in other words, presumably C and D modify each other in some way. So one of these becomes E and one of these becomes F. F is just a plain old intermediate. It's identifiable, but it cheerfully moves on to react another reaction. Okay? And it reacts with A. Don't lose track of this now. So A, as it wears many hats, an important food product. A reacts with E to produce G, which is waste. Now I know this seems bizarre to have a set of reactions that produce waste, but I want to emphasize to you that all living things have to do that. Otherwise, this kind of junk builds up in the system. So one thing that you do is, this is a way of getting rid of, let's say CND. If CND just build up in your body, it's bad for you. So an example of this would be carbon dioxide. Okay, as a metabolic byproduct, your body produces, when it burns glucose, let's say, sugar products, it produces carbon dioxide and water as products. Well, water is innocuous. Okay, water, your body has many ways of getting rid of water, your blood's full of water, so nobody cares. But carbon dioxide will kill you if it builds up in your cells. So what has to happen is, the carbon dioxide has to make its way to a point where it can get out of the body. Okay, and the primary way it does that is, it dissolves in the cell in water. It turns into something called carbonic acid. Carbonic acid is perfectly soluble. It travels through your bloodstream till it hits the liver. Till it hits the lungs, and when it's in the lungs, it breaks down back into carbon dioxide and water and is released. So the carbon dioxide goes away, the water just hangs around. Now, remember, this is where human beings are very teleological. We like things to have reasons. We tend to explain nature in terms of, spirits and winds or ghosts or something, okay? Something that has a purpose. In this view of life, this system simply persists. That's all it does. Why does it live? Because that's what it does. The purpose of life is to live, okay? And this thing will keep going as long as you keep supplying A and B. And you have a way to get rid of G. So that's all they mean by an autocatalytic system. The most primitive cells look like this. I mean, they're more complicated. This is the simplified version, but this is what you have. Now, I dread even saying any questions about that, but feel free to ask the broadest of questions about this.
Okay, broadest of questions about that, guys? The broadest of questions about that. This is a class of very broad questions. Does it make sense generally?
Does this make any sense? You're bothered by this? It offends your sensibilities?
And are there some things that you think are notable if we're going to make some parallels between a catalytic reaction system in a cell and a social system later? Can you?
Also want that you could almost think of this in many ways. You could reinterpret this entire diagram as an ecosystem.
Yeah.
In other words, molecules act as little entities that run within the cell at the microscopic level. Cells make life forms. Cells themselves can then clump together in this fashion to form different kinds of tissues they can differentiate. That forms an organism. Organisms can clump together in a kind of exchange of energy in something like this to form an ecosystem.
Well, so
I'll leave it to Lauren to talk about the transition from an ecosystem to a civilization.
Right, or social systems. But I mean, in an ecosystem, you have waste, right? And you have sort of energy sources. I'm thinking of the Isle of the Bunnies. I love thinking about those precious little bunnies getting with those, what was that, a wildcat galloping behind it in the snow there? That was grisly footage.
Lynx.
A lynx. But so if you've got too many bunnies, that's sort of waste, I suppose, or is it? I don't know. Maybe I'm thinking about something else. But I'm thinking about how one of my main things I'm thinking about is how the food source is a food source at one point. It's also a catalyst at another point, and we have to account for its waste as well. Is that right?
Well, don't forget, like for example, scurvy. Okay, scurvy was the disease of sailors because they were separated for a long time from fresh food sources. And something they couldn't get were fresh vegetables because they were difficult to preserve, given the technology of that time. What they were getting from fruit was obviously vitamin C. Vitamin C is a coenzyme. Okay, we directly intake it, and we can't produce it. Human beings and guinea pigs. are the only two animals we know of that don't produce vitamin C on their own, mammals. And matter of fact, that's one of the reasons we think of guinea pigs is it has almost become an archetype for a lab animal, because all the experiments to discover the nature of scurvy had to be performed on guinea pigs because they were the only other animal that got scurvy.
Mm.
Okay. Let's move on here. Oops. Sorry. Don't do that. So the idea here is this is how you think about something working. Are you still there?
Yep, we're here.
Okay. This is how something works at a microscopic level. Let's take it up to the ecology level now and start talking about, instead of A's and B's and C's being molecules, I'm going to skip the molecules organizing themselves into organisms. Let's go straight to how the organisms interact and make an ecosystem.
Okay, can I ask one question,
So,
Steve?
for example,
Can I ask
Sure.
one question? Because we're about to go from chemistry to biology. And so before we leave chemistry, I just want to say maybe we can talk about this on Thursday, but I'm just going to put it out there for you because I know we're going to be talking about complexity on the bigger scale. But I'm not sure if it was in that article that I was reading this morning or something else that I was reading this morning about how, I guess it wasn't, but it was something else. More complex molecules have more entropy than simpler molecules, is what was stated. Does that make sense to you? Okay.
Sure. I mean, look, a big molecule is going to have more available microstates than a small molecule. But on the other hand, think about it, a methane molecule, a tiny little molecule like methane, basically has a shape and it never changes, so it has very few microstates. A big, complicated, spongy protein is going to have a lot of different microstates. On the other hand, the protein, however, can act as an engine. A protein, by being complex in itself, can organize energy. So, I mean, it's, again, the key change we're going to make on Thursday is going from entropy to free energy.
Okay, good.
If you will, entropy is the evil twin. It's the energy you're losing.
Right.
And it's being degraded. Free energy is the energy that's available to do work.
Right.
The sum of the two is the total energy you have.
Right. Okay. Well, thanks. I'm looking forward to Thursday, but this is important too. So, go ahead.
Let's just go through a little bit of population stuff here. So, for example, this is what a population looks like versus time. This is a real population. These are midges. Midges are those annoying little flies that bite you. And they're indigenous to swampy areas. I've heard a few of those down there in Louisiana. And if you just plot their population versus time, okay, you see these kind of fluctuations, okay? It's just hard to predict what they're going to be doing, okay? So this is what the kind of data we have looks like. But the populations do go up and down. It's with generations. Now notice, this is reproductive generation. Okay, so this is roughly, the time intervals are roughly how long it takes for a midge to reproduce. Notice that you don't have any idea how long this takes. Is this 2 weeks? Is this a year? Is this 2 hours? Okay, so that's, one reason when you see these kind of graphs, understand that, look at that axis to see if it's actually time or generations. Now, here's interacting populations. This previous one was an isolated population, the midge population. Nothing really, there's no real predator-prey issues here. Okay, and it just goes up and down on the cone. Now, here's a predator and prey issue, okay? There's the prey, and there's the predator, okay? And one of these are bugs, okay, I'm going to generically call. Remember, you're dealing with chemistry, not biology. And if you really want to feel disgusted, this is adults per 12 grams of wheat. So deal with it. You've eaten these creatures on a regular basis most of your life, in flour-based products like pasta or bread. Anyone that thinks that most of your food, especially wheat, is free of insects, is living in a fantasy land. There's no way we can get all the insects out of the wheat we eat. So if you want a particularly disgusting concept to think of, 4 rat droppings are allowed per loaf of bread by the federal government. So enjoy. Okay, but notice. Okay. Notice, by the way, that this is the predator and that's the prey. There's always more predators than more prey than predator. Prey populations are much larger than predator populations. To give you an example, in the Serengeti, okay, you have about 5,000 lions, and you have about 1 million Thompson gazelles. Because you need that kind of ratio. You can't have predators and prey in roughly equal numbers. The prey have to be present in much greater amount than the predators. Okay, this gets even better. Here is an example of a bunch of populations of what are called exploited resources. In other words, these are fish that we eat, okay? And we are a significant predator for this population. We can easily hunt these things into extinction. So this is a plotted from 1950 to about 2010, and 2005 rather. And you can see these various populations and how they fluctuated. And if you look at, like, for example, this one here, this is the haddock, okay? Had a big nadir here, spiked up, went down, so on and so forth. Now, this may, of course, affect your culinary habits in terms of what's available, but you also got to realize that you're a fisherman, okay? Imagine trying to make a living if this is what happens, okay? You're seeing these small fluctuations, but they slowly slide, and then they slowly grow back, and they slowly die off, and they go back. And a real question is this: is this natural? Is this what happens? Populations naturally do this even when we're not involved, okay? And this is a managed resource. We're very careful with the amount of harvesting we allow because we're trying to keep the fish populations robust. Our ability to understand how these things respond to us and to other things, other shocks, changes in the environment, for example, is critical for the industry here. Now, again, it's an interesting problem, okay? This was, again, the realization, going back to our thinking about the island of bunnies. Early on, it was thought that, if you had a complicated system showing complex behavior, it had to be a complex system. May, a biologist, was one of the first people that showed that logistic map equation would produce very complicated behavior. And that's not a complicated system. That's not a complex system. It's a simple equation, but it produces complex results. And that confused everyone. That's why we give it the special name chaos. Chaos means it is basically a complex result being produced by a simple system.
But it's because of...
all of a sudden, this just messed things up.
But it's because you can only see that after many iterations of the system, right? So where you get to see
sure.
the sensitivity to initial conditions.
You need to look long term to see something.
Yeah.
You need to look long. You can't see this short term. All you would see is something rising or falling, and for all you know, it's starting to oscillate in here. But then you realize it's never repeating itself, really.
Right. This happened. I'm sorry, Steve. This happened about the same time, 1974, with May, as Lorenz and the Strange Tractor. Yes.
10 years later.
Oh, okay. Lorenz was
10
later.
years later.
Okay.
Lorenz and May probably knew nothing about each other.
Huh.
Especially in those days, meteorologists and biologists spent a lot of time talking to each other. And remember, no internet. We only had paper journals. So what journal did you march over to the library and read?
Yeah, we liked it
that was a big set. How many journals did you personally have delivered to your office? That was probably a smaller number. So people just weren't as casually aware of what's going on in other disciplines as we are today. And it's not like we're talking about Roman times, we're talking about, 50 years ago. Okay, now this is a big problem biologists deal with. I call it chaos versus stochasticity. Wow, there's a word, huh? Is the process being governed by deterministic chaos, which is a complicated result being produced by a simple system, or is it literally stochastic, which is a fancy word for random? In other words, it's literally random processes going on here. Coins are being flipped, okay? And it's very difficult to understand that. Another problem we have with studying biological systems is the problem of noise. Noise is the name we give to randomness entering the system from the environment. Okay? Now, it doesn't matter. The surroundings will produce noise, whether that noise is chaotic in nature or stochastic in nature. But from the system's perspective, it is very difficult to separate out chaotic behavior that I am having from chaotic behavior that's basically I'm getting bumped. I'm not, it's one of those things where if you're, usually, but here's a good college example, if you're drunk and you're staggering, you're walking back and forth in a kind of random pattern. On the other hand, if you're a perfectly sober person, but you're trying to fight your way through a very dense crowd, you may actually look like you're doing pretty much the same thing. But in the first case, it's your problem, okay? That's the example of pure chaos. In the second case, that's noise. You were trying to walk a controlled trajectory, you just couldn't do it because your environment was beating on you. And this is really hard to do. Just to show you one of the strange results that happened, one of the most studied insects is something called the flower beetle, which is a little insect, obviously likes to live in flower, and eat flower. But one of the interesting problems with studying the flower beetle, and we like it, it reproduces quickly, but one of the interesting problems is virtually everyone who ever studies flower beetles ends up allergic to flower. Okay?
Huh.
And that discourages people from pursuing these kind of studies. Biology is always trickier. Chemists can work in a hood in the lab, biologists have to go out into the field and deal with the problems that occur out there, and they can't always isolate their systems the way a chemist can.
So
there's a picture of another guy.
And so Steve, I just want to point out, what you just said about chemists being able to control their environment more than biologists. Physicists can control it still more than chemists, right, in a sense, right? That's sort of the hierarchy of this. Yeah.
Exactly.
Mm-hmm.
In terms of scientists, whether or not you want to put the mathematicians in there,
yeah,
it's a matter entirely.
Yeah.
This guy, by the way, we're going to talk about this other one in a minute, but this is the guy, Vito Volterra. He's a rather dashing fellow here. He studied fish populations, okay, and was able to model predators and prey populations of fish. Again, not ancient times, he's 20th century. And he was the first person to have any understanding of how these fish populations work. Remember, once upon a time, we just went to the shore and fished. We never gave a whole lot of thought to what was going on with the fish. As we got better and better at fishing, suddenly we had to take into account that we were perturbing these populations, and we had to understand them. Otherwise, we could just basically fish these populations into extinction, and that's been done. Okay. Now, here is the notion. So we're going to emphasize now, look at the most basic interaction in a population, the predator-prey interaction. Okay? A prey is typically an herbivore. It lives by eating the plants. Now, by the way, I want to point out, the herbivore and the plant are also a predator-prey population, but we won't get into that for now. Predators are normally animals that eat other animals, and they're meat eaters. So we have carnivores and herbivores. Other ways you can do this, by the way, is, example, prey could be human beings, predators could be a virus or a bacterial strain, a disease. There was one particularly amusing set of simulations that came out of the National Institute of Health. It was a population consisting of vampires, zombies, and human beings. So you had human beings as the prey. Vampires and zombies represented two different types of predators. A so-called smart predator and a so-called dumb predator that would drive the population to extinction. Everybody wondered about this and thought it was pretty funny until they realized they were actually modeling two diseases competing over a human population. Okay, so let's see how these guys interact. Predators can overrun the prey population. In other words, they can eat too many of the prey, driving the prey to low abundance. Low prey abundance, in turn, leads to a decline in predator population. If you have to eat these little guys to live, and you overeat them, and there aren't many left, then you start to die. The thing is, though, if you have a decline in the predator population, the prey population recovers, and it goes big again, which means the predator population can follow suit, and so on and so forth. And you've got a cycle with a well-defined lag built in. So here's our great picture again. I've used this picture before, but here's again. Here's the lynx, and there's the snowshoe hare. And, this is a race for dinner. Okay, clearly. All right, whether when he's going to be caught or not. And here are the populations. The victim, prey population, okay, has the two terms in this equation. So look, his rate depends on his own population. That's a straight exponential growth equation. So what that means is this population will either grow or decline depending on the value of R. So this is written as a growth, so the population is capable of doing that. Now, this is what we call a cross term. It depends on the population of the predator and the population of the victim. By the way, in case you haven't realized this, the reason we use victim is because predator and prey have both start with P. So notice that this represents the interaction of predator and prey. And you'll notice that this has a negative sign, which means that the prey population starts to decrease the more the predator and prey interact. Because that's bad. The prey interact with the predator. On the other hand, here's the predator. Notice that if we ignore the interaction term, this guy has a negative growth rate. So in other words, without interacting with the prey, the population of predator just goes away. The slope of predator versus time is negative. On the other hand, if predator interacts with prey, that causes a positive growth rate in the predator population. These equations are called the Lacta-Volterra equations. Okay, and they're some of the most studied equations in both social and natural science. These have been used to model everything from bunny populations to chemical reactions to organized crime. All right, you're going to like this one. In organized crime, the prey are local businesses, and the predators are mobsters, if you will, that extort money from these businesses. It's like they call it protection money where I'm from, because what it was, you were protecting them from the very people you were paying the money to. We don't want anything to happen to your business. So that's been studied, and these models reflect that very well. And obviously, for example, if the criminals get a little bit greedy, the businesses start to fail or move away. And of course, if they move away, then the predator population, the mob, has to get smaller because there's no money to support them, and then the business population will come back. So these are very common equations. And here's the kind of oscillations you get. This is the prey, and this is the predator. And notice the predator follows the prey population. So if prey grow in, the predator starts to grow, but then he starts to overeat. There's too many predators. This population starts to crash. That causes this population to crash. There's always a lag time because the prey has to respond to the predator. And notice one curve follows the other. Now, Does this work? Okay, this is a fascinating thing. Why do we like the hare and the lynx? Because of the Hudson Bay Company trapper pelt data. Okay, it's hard to get long-term data in biological systems, but this one works great because we've been trapping both of these creatures, but especially the snowshoe hare, but both for their fur. And the data goes back to 1845. So we assume, and this is an assumption we're making, that the actual populations are reflective of how successful the trappers were in catching them. And you'll notice that the data looks a bit noisy, but you definitely see this kind of notion right through here, for example. You see the predator following the prey population. And this is not uncommon, although there's a lot of noise in here, because of course these two things aren't isolated. There are other things eating the bunnies, and there are other things the lynxes can eat. Still, you're getting lots of Voltaire kind of behavior. And this is, again, this is why we like this particular example, because we get a lot of data. Now, in case you're curious, can we get better data? Absolutely. If we can make these things to microbes, or to an insect population, a microbe, those have many, many more generations over time. So you can actually study many, many more generations as a function of time. Okay, so the question is, how realistic is this? Problems. Unbounded victim populations grow in the absence of predator. Okay. Yeah, right. Okay, in the absence of predators, you take the predator away, the victim population won't grow without bound. I mean, you're going to run out of food. So that's the problem we have. We're not taking into account, if you remember the phrase, the carrying capacity of the environment for the predator, for the prey, for the victims. Notice this is a single prey specialist predator. Okay, in other words, all this predator can do is eat that prey. So this is, the lynx that can only eat snowshoe hares. A nice fat juicy squirrel is ignored. Okay? Very few predators are that specialized because they simply can't survive. If you wanted a great example of this, the Irish potato famine. The Irish became unusually dependent on one crop, the potato. So a single blight crashed their food supply system, and 2/3 of their population had to move. Okay, I mean, if you look at that time, 2/3 of I think Ireland was up to about 6 million people, and about 4 million of them, or something like that, immigrated into the United States. So literally 2/3 of their population went to another country to deal with that problem. The 2 million stayed because by that point, potatoes that were still surviving were sufficient to support 1/3 of that population. The other problem is consumption rate of predators relative to prey abundance is unrealistic. What the hell does that mean? Okay, it means that the predators stop eating after a while. Okay, in other words, if there's twice as many bunnies out there, doesn't mean you eat twice as much. Okay. This is called the functional response. It's the response of the predator to the prey population. Okay, if the prey gets too big, you don't always get the predator. The predators don't just necessarily eat more. Now here's a great example of that. In Africa, the wildebeest, okay, all reproduce at the same time. They have all their little baby wildebeest right around the same time. Why? The lions are the primary predator for wildebeest, but a lion doesn't like to take on a full-blown wildebeest, a full-grown one, because they're really big. So lions tend to eat little ones, the babies. So first of all, if they all go together, if they all give birth together, there will be afterbirth. Okay, the placentas, okay, will be just expelled from uterus of the female wildebeest. So there's basically a lot of fresh meat just lying around on the ground. And the lions will eat that, and it will satiate them for a short time. This gives the baby wildebeest a little time to get on their feet. Then, and this is the brilliant strategy of it all, if the lions can go after the little wildebeest, but they can't eat them all. They can only eat so many, and because there's a bunch all available at once, enough of them tend to survive. Notice that they spaced out eating the wildebeest. If they spaced out having the little wildebeest, the lions could eat a much higher percentage of the population at all times. So this has been observed. as an effect on a real population. So, just another figure of this, okay? Now, notice I can do a phase-space plotter. If instead of plotting, you know, predator and prey versus time, I can plot predator versus prey. So notice that here's the kind of picture we get as the prey population increases, the predator population will start to increase, but when it increases, the prey population starts to go the other way and decrease, so you get a spiral. Okay? Now, this particular set of parameters I chose here, this spirals out of control. And the problem when it spirals out of control is something starts to go extinct. Okay, so notice the predators get dangerously low. And of course, a real ecosystem can't slip down to fractions. I mean, eventually you go between having 2 left to having none left. Now, one of the interesting things about this is trying to analyze where this is stable. And what we look for literally is this. Where do the time rate of change of predators and prey go to 0? Because any place where the victim population isn't changing, it's had a steady state, the derivative will be 0 with respect to time. If we can find a place where both of these occur simultaneously, both are equal to 0, that means both populations are fixed and we have an absolutely stable ecosystem. Okay, so I go through a bunch of math, and I get this. Now, what are these stupid little arrows? This is an interesting thing to think about, and I don't think I have a picture here inside of that. Previous. Okay. This is one of the lockable pair equations. Okay, and that's the other down here. So this is the rate of change of the predator, PDT, dt, the rate of change of the prey. Sorry, I flipped that around, predator and prey. So what we do is we can calculate a little bit, which is literally the change in both populations. So they take that little arrow right there. The change in this direction is giving you the change in predator, and the change in this direction is giving you the change, change in prey. So we can, at every point in this phase space, just literally evaluate those 2 derivatives and plot this. Now, even if you don't understand this, you probably can figure out arrows, okay? So this is what's called a vector field, or better, I like this term better, a flow. Because that's exactly what it looks like. If I were to drop a little cork right there, your eye would tell you that that cork's going to do this. It's going to cycle around that way. Okay? Because your eye can just see it. Now, obviously, by the way, I let the plot go into the negative numbers down here. This is not biologically reasonable numbers. Given that we're interpreting this as a population, we can't have negative populations. So what you have here is the blockable pair system naturally predicts cycles. Okay, it doesn't predict steady populations. It predicts cycles. Because there may be a fixed point right there, but it's not a stable one. Now, to give you an example of what that looks like, here are some various classifications of fixed points, okay, and what happens around them. Blockable pair tends to produce what's called a center. A center is an unstable fixed point such that the trajectories that start around it tend to cycle, so we get those kind of oscillatory behavior we saw before. Now, if you move away, you can get 2 kinds of spirals. A spiral is, rather than just oscillate or circulate around that point, you either spiral away from it, or, into it, or away from it. Okay? So in this case, the populations both grow without bound. In this case, both populations die. Okay? These are various possibilities. These others, we're not going to worry about that. They're basically sink nodes, source nodes. Okay, so the populations move in different directions around it. But these are the kind of things we're interested in. But notice we can analyze the kind of behavior these populations will have. They'll spiral out of control, they'll spiral into extinction, or they'll just have a nice steady cycles. Now, I'm just going to show you some pictures here. You don't have to worry about how I calculated these. You can take my word for it that if you was given a computer and a little bit of time, this is just no big deal to do. So this is, again, predator abundance predator population versus prey abundance on the horizontal axis. These are the little pieces of the vector field, okay? So here's a particular one. This population just cycles around here. Now, one thing I could do is this is the response of the prey to the predator, okay? This predator line is where the predator population is unchanging, so sorry, I'm doing that. This is where dV/dt is 0, and it's a line. This is where dP/dt. This is where dV/dt is 0. Okay, these are sometimes called isoclines. Who cares? All right? They're places where the individual populations of predator and prey would be stable. Where they intersect is where both populations would have a fixed value. Now, this is a more realistic representation of the prey response, okay? What it says is prey have a carrying capacity. They can't just grow without bound. There's a point at which they run out of food. If we simulate this population with bending that line over like that, then these two will spiral into a stable population. Okay? And this is not locked to Volterra. Notice I've done this by adding this term. Who cares why? Okay? You can visualize this. By bending this line down, it changes those little arrows such that this will spiral in this way. So notice you spiral in if you leave your predator isocline vertical, but you bend down your prey one, so your prey basically can go extinct if it goes beyond its food source. Okay? Now there's all sorts of things we can do to shape that prey response, okay? And these are just very different responses. Let me just show you what they look like. So this is the number of prey eaten by predator versus prey abundance. Type 1 is stupid. It says if the more bunnies we have, the more we're going to eat. So, some fox will, some lynx will suddenly start eating 52 bunnies an hour. That's not going to happen. These are two more normal responses. Namely, this little factor is called simply satiation. I'm full. Okay? This is a stable one, okay, a rather gradual one. This one is really interesting. This is called a sigmoidal type curve. In other words, what happens down here is the predators have a starvation mode. In other words, if the prey get rare, the predators can slip into this low-calorie mode and not eat much. But if the prey become common, they can slip into a high-calorie mode, where they can eat more and exist in the planet. Now, these are different ways you can have this response. Suppose this is the prey saturation. Notice that the predator satiation. What it does something different to the prey line. Okay, what it does, it points it upwards instead of downwards. And if you look what happens here, this population spirals out of control. And if you spiral out of control, you do real well, but eventually you crash, because eventually you hit 0 axis for prey, and then everybody dies. So what we can do instead is do both. This is the predator satiation part of the curve. This is the prey carrying capacity part of the curve. And I can now get stable cycles, okay, by simply putting it in the middle, all right? And this means predator and prey can reach a nice stable, it's a cycle, but it's a stable cycle. They continue back and forth. This, again, you're on this part of the curve, remember the downward part. These spiral into a nice stable population. Okay, so this means rather than stable cycles, you literally get stable populations of both. So predators are always at this number, prey are always at this number. If you end up on this part of the graph, then you get instability. This eventually leads to prey extinction, which kills the whole ecosystem. So these are different ways of understanding what can happen. Now, this leads to an interesting problem we're going to call the, okay, and so let's watch how this works. This is what the name is. If you actually are hitting right at the top of that curve, okay, that's the Volterra system. This Okay, well, you're intersecting on the downward range and getting a stable population of both are called an inefficient predator. Now, in this case, inefficient is good. Inefficient means this thing will not eat its population to extinction. This, on the other hand, is the intersects on the upswing side. This will spiral away until eventually the prey are dead. This is called an efficient predator. And an efficient predator is a bad thing because it will simply eat its prey into extinction. Now, here's a fascinating problem that they realized. This is a nice stable population, right? There's the prey carrying capacity, all is good. What if you go in and all of a sudden the prey have a new food source? So somebody introduces a new food source such that the prey carrying capacity increases. Okay, so bang, look at that. Boy, lots of prey, right? Lots of good food, we're doing great. Look what happened. I went from intersecting it at the stable area to intersecting it at the efficient predator area. Now, by increasing the food for the prey are driven to extinction. This is called the paradox of enrichment. Okay, you can actually make the world better for prey and therefore kill them off. This is a real interesting problem. Why? Well, here's interesting examples of this, okay? This is mussels and starfish, okay? Now this is, you normally think of the predator-prey population, the running animal, the cat chasing it, okay? Here's the slow motion version. Mussels, which are not the fastest animals in the world, can be overtaken by starfish. Okay, so you can imagine it's not going to be a cable show anytime soon. But notice what happens, okay? In a normal situation, you have a small predator population and a big prey population, which tends to be stable. The minute they started to subsidize, in other words, make life better for the mussels so they could harvest more of them, they drove them to near extinction. Okay, because what happened is by making an exponentially more amount of mussels, the starfish blossomed, okay, and became an incredibly efficient predator. And even when the mussels started to be brought back down by an increased predator population, the predators didn't die off quickly enough, and they continued to eat the mussels into extinction. So this is one of the best examples of this. Here are some others. Okay, this was a pure lab thing. They had a mite and a mite predator. I notice I don't call it what it is. The guy just literally fed the mites more and managed to crash the system. There's another laboratory system where it was algae and zooplankton. So algae with the food, it's a plant, zooplankton is an animal that eats the algae. They were able to crash it by adding nutrients to the system for the algae. Pest outbreaks and crop failures. Now this is really an interesting problem. If you start growing more and more and more crops, okay, and of course now increasing carrying capacity is a farmer helping the crops to grow, you suddenly allow the predators for the crops, the pests, to suddenly spiral out of control and eat all the crops. Okay, and of course you just had my example of the mussels and starfish. So we don't know. Okay, and one of the reasons there's still other features we have to model. Prey refuge. Okay, in any natural system, like rabbits have holes. So they can get into their hole and the predator can't touch them. You have to take that into account. You have competition between predators. Okay, rarely do you have a single predator. You have spatial dynamics. In other words, the prey can say, I'm sick of this, they can move away. This is the example of my business example, my business and the mob, okay? If the business is simply moved to another city, okay? Here's an example, this turning, instead of having the hump, this turns back up down here. This is an effect of prey refuge. In other words, there's a place the prey can hide. They start to get, their population gets too low. Notice this restabilizes the cycles. Okay, so these are just complicated systems to try to model. Now, we can think of it this way. most species are described by either cycles or chaotic cycles, i.e. stranger attractors. Okay, they're still cyclish things, so whether it be a molecule or a bunny, your population is probably being described by a cycle. Those are the most common things we see. You don't see fixed points as much.
And Steve,
cycles converge.
Steve, can you hear me? Is that that's just population levels?
Yeah.
Yeah.
Population levels. Now, if you're talking molecules, you're talking about concentrations. But concentration is just a molecule population.
Right.
So most populations go up and down in cycles. Chaotic ones or regular ones, but still cycles. Just like the example I had from cellular biochemistry, right at the beginning, okay? These cycles can merge and interlock to form what are called hypercycles. In other words, interlocking cycles, almost like gears, so that one cycle drives another cycle over here, so you suddenly have lots of predators and prey in some sort of interlocking cycle. You can even have catalytic things, okay? Human beings can move into an area, force the prey into a smaller land space, where the predators can catch them faster. So human beings can be a catalyst. Now, a terrible example of this, I was on a safari once, and we were in little vans, and they wanted to show us a lion catching a prey, catching a gazelle. And so we're out there, and we have 5 little vans, and all of a sudden, these 6 lions appear and are walking between the vans. It was stunning, and all of a sudden I realized this was literally cooperation between the human beings and the lions, because the gazelles weren't afraid of the vans because they knew that they didn't think about the predators. But the sound and the smell of the vans shielded the lions. And all of a sudden the lion shot out from between the van and caught the gazelle. And it was really an hysterical situation of seeing the human beings being literally a catalyst for this particular reaction.
Yeah.
Ecosystems, again, can mimic autocatalytic cycles. Okay, you can do that kind of thing. Enzymes get replaced with a variety of things. Human beings in vans, human beings changing vegetation, geography. I have a place to hide, I don't have a place to hide. Weather can change, that'll change everything too. So you still get these kind of complex interconnecting cycles, okay, that are typical of living systems from the biochemical level straight up to the ecosystem level. And I'll let Lauren tell you if this persists to civilization level.
Yes, and indeed it does, as you help us catalyze these relationships ourselves. And I think that last...
So here it is.
Yeah, go ahead. Well, I was just going to say that last statement of ecosystems mimicking the structure of catalytic systems, autocatalytic systems, is especially good. Yeah, we ought to seize on that.
Okay.
Well, you're ending right on time, Stephen.
My plan on Thursday.
Yep. Go ahead.
Is to go through the Kazan papers.
Great. That's a real climax for us in natural science, I think, for this section of the course. I think that'll be great to go through that. All right, Stephen. Thanks, man. Appreciate it.
Okay. No problem. See you Thursday.
See you Thursday.
Have a good day.
Yep. So, guys, that'll be no readings for Thursday. We're just doing Chai Song. We'll go over that. Wish good luck to the MCAT people. Person. Godspeed. And hang in there, guys. We're gonna, you can see it. Sort of patterns are emerging. This is the natural science world, and we'll be moving into territory that's more familiar for a lot of us. Some of these people have had a lot of this, so it's old school for them. Yeah, go get them tomorrow. Thank you. Not a bit. Just focus. Yeah.